Okay, so what we're going to do in this lecture is sort of follow along from one of the ideas we left with at the end of the first lecture, and that has to do with the question of model selection, and ultimately this concept that we will talk a fair bit about called multi-model inference. So here we are, model selection and multi-model inference, and as you'll see on the screen here, without much pain. So some of this stuff can get pretty technical. The basic ideas are relatively simple, and so I'm hoping we can try to find a balance between, full complexity, full understanding, full details, which are, not always the easiest, versus just a superficial treatment, which really doesn't give you enough to work from. So without much pain. So a little bit in the middle, hopefully. Okay, so we start from a simple premise that the hypotheses that we're interested in testing with regards to any kind of biological system, like, do young animals have a different survival rate than old animals, is a hypothesis. And we express those hypotheses in the form of models. And specifically for this class, the idea of statistical models. And the thing about a model that we have to keep in mind is that for any given model, structure will yield estimates of parameters. All right. So as a result, we have to understand that each model is going to potentially yield different estimates of parameters. And in fact, can almost be guaranteed to do so, except in sort of a very few special cases. All right. So each model is a representation of a different hypothesis. The models themselves will yield different estimates of the parameters, and those estimates will differ amongst the models. And so given that a given model structure represents a single hypothesis, and you might have multiple sort of ideas about the way that you think the world works, how do you determine which model structure is best? And then, as is sort of indicated parenthetically here, well, what do we mean by best? All right, so models are hypotheses. All models are wrong and some are useful. So we understand, or hopefully we understand from courses where we've been exposed to the idea of models, is that all models are representations of some unknown truth. They're basically cartoons, and they're largely conditional upon the available data. And it probably goes without saying, but I'll say it anyway, that small data sets cannot support complicated models. And so since models are cartoons or sort of representations of reality, and since none of them are, truth, they're all sort of representations or approximations to truth, how do you know which model to use? And so the idea of selecting amongst models is pretty clearly important, and it's an area that is getting a lot of sort of interest and research at the moment. When you sort of play with models and data, they're often two, not mutually exclusive, but it's just convenient to think of them this way, objectives. One of which is to come up with the best estimate of a parameter. So if you're estimating abundance, you're estimating that parameter, and that's what you're interested in. Sometimes, though, the purpose of fitting models to data is not simply to estimate parameters, but it's to learn something about biology. You may be interested in whether male and females of some species have different survival probabilities, and the actual probabilities of themselves may be of less interest than the general question, do males and females differ? So sometimes we're interested in the best estimate of the parameter because it's the parameter estimate itself that's relevant, and sometimes we're simply trying to estimate something to learn something about the biology, and often we're doing the two simultaneously. So you can think of these as sort of points on this continuum based on the objective of fitting models to the data in the first place. All right, so last time we had a look at one of these sort of fate diagrams. So this was a simple case where you imagine a closed population of organisms, and you grab a sample. Some of them you grab, some of them you don't grab, or seen or not seen. And then with some probability, you see them again or you don't. All right, so in this particular model, as you recall, we have a subscript here, 1 or 2, representing the particular time occasion for the sample. So P sub 1 is the probability that the first time you reach into the population that you encounter or grab or see the individual. 1 minus P1 is the probability you don't. P2 is the probability that you detect them on the second sampling occasion or you don't. 1 minus P2, the probability that you don't, with the corresponding encounter histories and, the probability expressions that go along with those. Okay, well, that's one model. And again, the point that we made last class is that by subscripting for each time step, we're allowing for the detection probability for the first sampling occasion to potentially be different than the second occasion. So since those occasions are discrete points in time, we would potentially call this a model where P, the detection probability or encounter probability, is time dependent. Well, here's a competing model. We might believe that instead of the encounter probability changing over time, that in fact it's constant over time. So no subscript. So the probability of detecting the animal on the first occasion, we might imagine or hypothesize, is the same as the probability of detection on the second occasion. So this is a model that we might call constant over time, sometimes known in the vernacular as a dot model. Okay, and you'll sort of get a better sense of why we call it that as we get into this more. Okay, but we have two models, one where P varies over time and one where P doesn't. So why is that a big deal? Well, as we talked about last time, it doesn't change the basic encounter probability histories, but it changes the probability expression underneath it. So for a given set of data, we're estimating different parameters, and in the context of abundance estimation, which is how we set up that particular example, we get different numbers. Right? So when we had time varying encounter probabilities, we had an estimate of abundance of 100. When we had time constant or the dot model for encounter probability, we got an estimate of abundance for the data that we were working with of 108. And it's a reasonable question to know which of these two values, which are different, should we have the most faith in. All right, so because the models that we're building represent different hypotheses, then the number of models that you have can potentially be very large. In the sort of 60s and 70s, if you were a student and you wanted to be one of the cool, sort of with it, well-read people, this was a book that was on a lot of shelves, Persig's book on the Zen and the Art of Motorcycle Maintenance. And one of the quotes from that, which gets used with some frequency, is that the number of rational hypotheses that can explain any given phenomena is infinite. Well, that's a little obtuse, so let's just talk about it in the context of phenomena that you're familiar with. We know that, for example, there's lots of evidence empirically that the global temperatures are increasing, right? So global climate change or global warming, if you really want to simplify it down a bit. So why is that? Well, just think of all the different hypotheses that you could potentially come up with to explain the phenomena being the increase in temperature. Well, maybe it's because of CO2. Maybe it's because of CO2 and sulfur dioxide. Maybe it's because of just sulfur dioxide. Maybe it's because of CO2, sulfur dioxide, and increasing number of sunspots on the sun. You know, there's all sorts of things that you could do, basically being that the number of rational hypotheses that can explain any given phenomena is infinite. Now, what's really important to mention here, and it sounds like a bit of an aside, but it's not, is most of the time in ecology, or frequently in ecology or climatology or, certain types of science, we're not really doing experiments. So when you do an experiment, the number of factors that are relevant for the variable that you're interested in, the response variable, let's say ambient temperature, is small because you're controlling that as the person doing the experiment. Right? So if you're looking, for example, at the amount of, or the height of a plant above the ground, so above ground biomass, you believe that maybe it's a function of the amount of nitrogen in the soil, so you do an experiment where you have 0 nitrogen, 5 parts per million, 10 parts per million. You do a set of controlled plant experiments, at which point you've specified the hypotheses, and therefore you've specified the models. On the other hand, if you're not doing an experiment, you're simply watching a system, then this is where Persig's quote becomes particularly relevant. The number of rational hypotheses that you could come up with for any phenomena that you're observing in nature, if you simply watch something through time, is extremely large. You haven't done the experiment, and so therefore you need to think a little bit about, well, what models do you fit? And each model potentially gives you a different answer. So how do you sort of choose amongst the different models? In the experimental context, you might have the experimental hypothesis and the null hypothesis. But if you're watching a system, let's say climate change, you don't actually have a control. You don't have a treatment. You just got lots of different hypotheses, lots of different stories that you might propose to explain the phenomena. So how do you select amongst those models? How do you use the information over the set of models that you might try fitting to some hypothesis? And that's really what we're going to talk about here, because that's by a large stretch, the majority of the situations you're likely to run into. We don't do a lot of experiments in ecology, largely because it's hard to scale them, but we do lots of storytelling based upon the time series of information that we collect. We follow something over time, and then we try to tell a good story, and one of the main tools that we use to tell those stories is models. But which model is right? Which is the right story to tell? So for the abundance estimation, again, something that we talked a little bit about last class is that there's a fairly large set of models that you can build right out of the box. Here's 8 of them. These are what I call the sort of standard 8 abundance models that differ based upon whether you believe that things are time dependent, constant over time, maybe functions of the behavior of the animal pre and post capture. Maybe every animal is flipping different coins or combinations of those things. So there's a whole different set of classes of models. Each of them you might think is reasonable to at least consider, but every one of those models is going to potentially yield a very different estimate of abundance. So which estimate to use? Which model do we want to think about? Which sort of set of models are the reasonable ones to try? And that's the kind of thing that we're faced with in this lecture, and largely we're going to face going forward in this course. Now, there is a classical approach to doing this, and it does relate to this idea of experiments and null hypotheses and so on. And you absolutely need to be aware of it because you need to be able to understand it enough to contrast it with some of the other approaches that we're going to talk about. So one of the classical approaches to doing sort of model testing in statistics generally is something called the likelihood ratio test. Now, the reason I want to focus in on this is, other than the fact that it's pretty fundamental, it underlies an awful lot of the statistical tests that you do, even though you may not know them as likelihood ratio tests, but it relates directly to the maximum likelihood stuff that we developed in last lecture. So suppose that you've got some likelihood function, and that's what we did talk about a fair bit last time, is you can derive these likelihood functions, the likelihood of a parameter value or set of parameters given the data. You derive the maximum likelihood estimates for the parameters. Okay, so that's one model. And then maybe you fix some of the parameters to specific values and you maximize the likelihood with respect to those. Okay, so let's call this particular model, the second model, a restricted likelihood. All right, so in the first model, you got lots of different parameters. Let's say time-dependent parameters. And then in a second model, you restrict them. Maybe you make them constant over time. And so we'll call this restricted model little r, and therefore the likelihood for that restricted model little r is nested within the model where you allowed things to vary over time. Okay, now we'll get to what we mean by nestedness in a second. But okay, so we have 2 models, one of which, let's say, has full time variation in some parameter, and one where maybe you force it to be constant. Okay, now, if you take those 2 models, and each of those 2 models has a likelihood, okay, so the likelihood of what I'll call the full model with full time dependence, L sub f, and the likelihood with the restricted model, L sub little r. So 2 models, L sub f and little r, and you can think about one as being nested within the other. Now, we'll talk about nestedness in a second, I think, on the next slide. But given that you have these 2 models and they have this nested relationship with each other, this theory, the likelihood ratio theory and the test, says that the distribution of twice the negative log of the ratio of the 2 likelihoods is chi-square distributed. That's a lot of words. Okay, so here's really all you do. You take the likelihood, the actual value of the likelihood, not the parameter estimates, but the value of the likelihood evaluated at the MLE for the full model, let's say with time dependence, and the restricted model, let's say constant over time, and you take the ratio of those 2 things. Okay, if you take -2 times the log of that ratio, then that's distributed based on the theory, which we're not going to develop, we'll just appeal to, as a chi-square statistic with the degrees of freedom that's based upon the difference in the number of parameters. All right, so that's a mouthful, but basically what we're doing is we're saying, look, we have this thing called the likelihood. All right, and we've maximized it. And that's where the likelihood of the parameter given the data is at its maximum point. So one way to think of likelihood is good fit. The higher the likelihood, the better the model fits to the data. So we're looking at the ratio of the fits of the full model and a restricted model. Okay, And that ratio on the -2 log scale, we could show by some algebra, is chi-square distributed. And most of you have seen a chi-square test before, and you may recall that in a chi-square test, you have degrees of freedom. And where do you normally use chi-square tests? Well, you have a hypothesis that predicts that you should have a certain number of individuals in certain different classes, of individuals, so a frequency table, and you're comparing the observed frequencies and the predicted frequencies, and that difference is chi-square distributed with the degrees of freedom based upon essentially the number of classes of individuals you have. Well, here, the degrees of freedom is essentially the difference in the number of parameters, right? And we'll see that in a second. All right, so here's an example, and we're going to step through this example a little bit, and it'll make it clear what we mean by parameters and hopefully then what we mean by nestedness. So suppose you build some kind of linear statistical model which relates the variation in something of interest, let's call it theta, to two different sets of parameters. So let's just say that theta is a function of a couple of different things, and we'll just call those parameters alpha and beta. So theta, the thing we're interested in, is a function of alpha and beta. And let's say parameters alpha and beta can vary over some location. So the location we might index with I. So alpha I and beta I, or constant. So let's imagine that we have sort of 2 points on a continuum. Either alpha and beta vary over space, or they don't. And if they don't, they have a little dot notation underneath it, which is where the idea of a dot model comes from. So this dot subscript is relatively common and standard. So dot models, no variation over space or time. And then I usually means variation over space or time. Okay, so let's just make this really simple. Let's just focus in on alpha. All right, so we want to know if this parameter alpha varies over space, and let's say our locations are 1, 2, and 3, or it doesn't. Okay, so our null hypothesis is that potentially alpha 1 is different than alpha 2 is different than alpha 3. So this null hypothesis basically says that there is variation, some variation over space. If they were all equal to each other, then there is no variation over space. Okay, so here's our first model, our model with a hypothesis that says that alpha varies over space. So location 1, 2, and 3. So we have a separate parameter for each location. So that's our full model. And a restricted model says, no, alpha is the same everywhere. So it's simply an alpha dot model. So the betas, which we're not going to focus in here, we're basically going to say they differ over space, and it does so for both models. So what makes these two models different? The full model has 3 alpha parameters, 1 for each location. The restricted model has only 1 alpha parameter that's the same over all locations. So the idea then is that the second model, the little r model, is a subset or a special case of the first model. And that's the language that is in mind when you use the phrase nested. This model is nested within this model. So the little r model is nested within the F model because the little r model is the F model with a constraint or some condition put on it. It still has alpha, but the difference is now instead of alpha varying over time, it's nested within it. Okay? And that nestedness is important because what that is implied when we do this. So if model little r is nested within model F, then we can go ahead and use this likelihood ratio test. Now, what can we predict right away about these two models? So this is an important kind of conceptual point. When you fit a statistical model to data, there's lots of ways to sort of think about this, but one way, which is sort of a mental or visual heuristic, is to think of it as follows. A statistical model is like a ruler. You've got a bunch of data and you're trying to fit the model to the data. All right, so think of the data as some kind of shape, all right? So some weird kind of shape. And you've been asked to measure how long is the shape or what are the dimensions of the shape. So you have 2 choices of a ruler. You have a ruler that you can bend in lots of different directions. Okay? And so it's not hard to imagine that a ruler that you can bend in lots of directions is going to fit some uneven shape better than a ruler that only is straight edge. Okay? So if you have a ruler that you can bend in lots of different directions versus one that you can't bend, then it's pretty obvious that the ruler that you can bend is going to fit the data better. Okay, now, if it fits the data better, then how does that reflect in these things called the likelihoods? Well, on a -2 log likelihood scale, the model that fits the best is going to be the one that's closest to 0, because that means that the difference between what you predict and observe for the model is as small as possible. The smallest you could ever be is 0. So what would we predict then is that the likelihood for the full model, the model that's got the most parameters, each parameter is like a bend in the ruler, is going to fit the data better than the model with fewer parameters. So by definition, a model with more parameters is going to fit the data better than a model with less parameters. Okay? So the question becomes then, how much of a difference is there? Is that difference statistically significant? Is the difference in fit statistically significant? And that's what you're getting at here. If the fit between the reduced parameter model and the full model was identical, this would be 1, the log of that would be 0, and there is no statistical difference. So if this ratio is really big, that implies that there really is a difference in the fit, that adding more parameters really does significantly improve the fit, and you can measure that significance using a chi-square statistic. Okay, so let's go back to this. So again, the idea here is nestedness. If you just look at it for a second, you should hopefully see that L sub r, the likelihood for the small r model, this whole model is nested within this upper model. This upper model has full parameterization in the second model. The reduced parameter, restricted parameter model is essentially just a special case of the first model, the full model, where we've applied a constraint to the alpha parameter, that constraint being that they're all the same. So little r is nested within F. Okay, so the restricted model is nested within the full model. Now, if that nestedness occurs, then you can do a likelihood ratio test. Okay, so you fit you come up with a likelihood function for the data, you fit the model, and you come up with your not only your parameter estimates, which is what we focused on last lecture, but the actual likelihood itself. We do the exact same thing for the restricted parameter model. We get the two likelihoods, compare them, and we get our chi-square statistic. All right, and so the degrees of freedom, delta k, is simply the number of parameters that differs between the two. And that, if you do that, is a test of the null hypothesis about alpha. So if you find a hugely statistically significant difference between these two, that tells you that that better fit of the full model is important. Okay, if these two models basically fit the data equally well, so you get a non-significant chi-square, that says that you don't have much compelling evidence that alpha varies over space. If the difference is statistically significant, then you can make the argument that there is some spatial variation in alpha, and it's sufficiently big that, in fact, you see the difference in the fit. Okay, so just think of, again, the bendy ruler. If the surface you're trying to fit the ruler to is relatively flat, then whether you use the bendy ruler or the straight edge ruler isn't going to make much difference. But if the surface you're trying to fit the ruler to is really bumpy and it's got all sorts of weird hills and valleys and stuff, then a model which is flexible is going to do much better than a model that's a straight edge ruler. And so therefore, we might imagine that it's going to show that there's a significant difference between these two models, which you would then take potentially as evidence that alpha does vary over space. And that's it. So that's the test of the null hypothesis about spatial variation in alpha. Okay, well, let's move to an example that we are actually familiar with. We do the simple two-sample abundance test experiment that we introduced in the first couple of lectures. So recall, here's the likelihood expression for time-dependent P. So P1, P2. We had 28 individuals that we saw the first time and the second, 42 individuals that we saw the first time but not the second, 12 individuals we saw the second but not the first time. These are the probability expressions in the numerator for each of those encounter histories. The denominator, you'll recall, is the probability of being detected at least once. We grind through the algebra, get the computer to do the work. Here are the maximum likelihood estimates. P1 is 0.7, P2 is 0.4. Now, what we didn't focus in on, but we now do, is the following. How many parameters do we have? Well, we have 2 parameters. We're estimating P1 and P2. So n equals 2 parameters. What was the likelihood? Well, in the output, especially from the R script, but we also saw it, we could see it in the Maxima script. That's the likelihood. -2 log likelihood for this model fit to those data, 28, 42, and 12, the frequencies of the encounter histories, is 162.4964. All right, so keep that number in mind or write it down. So that's the likelihood given those data and given this model with time-dependent variation in P. So here's the same data fit to a model where we assume that P is a constant. So still 28, 42, 12, but the model equation is different. We run through the analysis. Now we only have 1 parameter. We're estimating P. But look at -2 times the log likelihood. It's 180. So again, remember a point I made a moment ago, that on the -2 log likelihood scale, the closer you are to 0, the better the fit. And we expect a model with more parameters to fit better. It's a bend to your ruler. So 2 parameters, 162, 1 parameter, 180. 162 is smaller, closer to 0 than 180. So right away, we see that the model with more parameters seems to fit the data better. But is that difference between 162.5 and 180.1 significant? Well, we can use this likelihood ratio test based on the chi-square. Again, there's the basic expression. Since it's logs, we can simply rewrite this, and sometimes it's convenient just to sort of do it this way, as simply the difference between the two, so -2 times the log of the numerator minus -2 times the log of the denominator. So that's how you can move things around for this fraction. We take the 2 likelihoods, -2 log likelihoods, so 180.1 minus 162 equals 17.6. Now, how many degrees of freedom? Well, again, the degrees of freedom is the difference in the number of parameters. So 2 parameters for the time varying model, 1 parameter for the time constant, so 2 minus 1 is 1. So if you fire up a chi-square test for a single degree of freedom with an alpha level of 0.05, so this is the magical 5% level for significance, that value would be 3.84. Well, our statistic is 17.652, which is way, way bigger than 3.84. So we would conclude, based on this, that there's a huge amount of statistical support in the usual sense for the model with time dependence rather than the model with no time dependence, meaning we might argue that we should have more faith in the model with time dependence, and therefore if we go all the way back, if we believe the model with time dependence is better supported by the data, then we should accept 108 as being closer to truth than 100. Okay, so that's sort of the gist of the idea. Okay. Now, let's look at another example, and then we're going to look at some computer code. Let's look at another example. So here we might be working with deer. So here's a male deer and a female deer, and we might have a biological interest in the question of whether male deer and female deer have different survival probabilities. Okay, so we do a study. We take 50 individuals from each sex, 50 males, 50 females, and we radio collar them so that we know for sure what happens to them. So this is analogous to the first question in the assignment that you have been working on. And then we would simply record how many survived. So we see that 20 out of 50 males survive and 35 out of 50 females survive. All right, so you look at these and you kind of go, okay, so what does that actually mean? Well, that means that we have 40% of the males have survived and 70% of the females have survived. And then the question is, well, is that a real difference? Okay. If you ignore the sex difference, then 55% of the total sample survive, pooling the sexes. So what you're interested in is whether or not you actually have evidence that males and females differ. And you can use the likelihood ratio test to get there from here. So let's see how that works. You start by building a model with no sex differences. And what kind of model is it? Well, it's binomial. You either live or you don't. Right? So we go back, 20 lived, 30 died. So it's binomial. So it's like a coin flip. Every animal flips a coin, live or die. 20 of them flipped the coin that said live. 30 of the males flipped the coin that said die. 35% of the females flipped the coin and said live, 15% or 15 of the females flipped the coin and said die. Okay, so it's a simple binomial likelihood problem. So here we start off with our model with no sex differences. Okay, so no sex differences, there's our simple equation. You could figure that out. We take the log of that, there's our log likelihood, and we can maximize it. We know exactly what to do. All right. And at the MLE, once we've maximized it, we take that MLE value and put it in here. We get the actual value of the log likelihood that we need for the likelihood ratio test. Now, our first little new bit here. For our second model, we have a model with sex differences. So how do you actually set that up? Well, the likelihood, because the males and females, we believe that everybody's fates are different, we can actually just multiply those together. So this first term is the binomial piece for the males, and the second term is the binomial piece for the females, right? And we simply put the two probabilities together, and we end up with our log likelihood expression down here below. So you pause the video and stare at this for a second, and you'll see, that it's a fairly straightforward sort of extension of what we've already done. Okay, so how do we actually do that? Well, let's go ahead and fire up some software to do that. All right, so I'm going to jump out to R, and we'll see how we do that. All right, so let me go ahead and I'll start R here, and I will open up the script for the two-sex problem. So this is a script that I will have sent you or will send you, and so just let me get into that. All right, so lectures, and here we are in lecture 2, and here's our code, here's our example demo, here's our binomial. So binomial sex problem. So there it is. And let me just have a quick look at this. Okay. So once again, we need to do some optimization. So I'm going to go ahead and run this to load the OptimX package. And I'm going to start by building the no sex model. So if we go back to our PowerPoint, here's the no sex model. It's a standard binomial likelihood problem. We're basically putting all the sexes together. So 55 out of 100. Again, so 55 lived, 45 died, and it's a simple version of the coin flip problem that we started with. So there's my function for the no sex model, and I simply call it F for function no sex. Okay, and what are the actual results? What are the parameter values? So I'm going to go ahead and run this, and again, this should start to look a little familiar. We did the same thing for the coin flip. I run that, and there's my results. So results no sex is what I called it. So let's just look at it. Results no sex, and remind ourselves what comes into the results. Well, there's the parameter value. There's my MLE, 0.55. Well, what else could it be? 55 out of 100 survived. So 0.55 divided by 100 is my MLE. What's my Hessian that I could take the negative inverse of to get the variance? Well, there it is at the bottom. But this other value, which we kind of blew past on the first lecture because we didn't really need it, that's the likelihood. That's the value of the likelihood at the MLE, okay? So that's the number that we need in order to construct our likelihood ratio test. All right, so now we build the model for the two sexes. So this is essentially the equivalent. This is the sex function based upon what you saw on the PowerPoint slide. I'm going to run that. I do the optimization here because I have two parameters, survival for males and survival for females. I'm using Nelder-Mead. Again, we talked a little bit about this in the first lecture. And I run this. And now I look at the results for that model, results for sex. There's my two parameter values. And there's my likelihood. All right, so the model with two sexes is the Bendier ruler. And so we anticipate it's going to have a value of the likelihood that's closer to 0. So negative 64 is closer to 0 than negative 68. So our intuition is met. There's our Hessian, which we could invert for the variance covariance matrix. But our focus here is mostly on the likelihoods. All right, so what do? Well, we're going to take the likelihood estimates for those things, and we're going to calculate the difference. So remember from the PowerPoint slide, what we need to do is work with the differences in -2 log likelihood. So it's the differences in those likelihoods that we actually need. Now, because it's -2 in the log likelihood, we can basically just get away by taking the actual difference by simply taking the results and one minus the other. So I'm going to go ahead and do that. So the difference in the likelihoods between these two is simply this value. It's 4.62. So the difference between negative 68 and then and down here negative 64 is 4.62. So the question then becomes, what is the difference between those two? What's the difference, and how does it compare to a chi-square? So we're going to make use of the built-in capabilities in R to calculate the probability of a chi-square. So we take the difference in those results, the difference between the two likelihoods. It's got 1 degree of freedom. So the model with sex has 2 parameters, male and female survival. The model without sex has 1 parameter, so the difference is 1. And we simply calculate the probability by using this term here. And there it is. So the likelihood ratio test of the difference gives us So 4.62 with 1 degree of freedom is marginally significant at the 0.5 significance level, 0.03 being slightly smaller than 0.05. So we would argue that there is at least some degree potentially of difference in survival between male and female deer. And that's it. And so if we actually go back into the PowerPoint for a second, that's exactly what we'll see. So here are the same basic values that we just saw on the thing. So the parameter estimate is 0.55. There's the variance, which we're not focused in on here. Here are the parameter estimates for the model with sex differences, 0.4 and 0.7, which is exactly what we expect them to be. Here's the likelihood estimate for the model without sex. Here's the model with sex. The difference between the two is 4.62. So again, this is exactly what the R script was spitting out. In fact, I just cut it and pasted from the results. Okay. So there is some evidence of a sex effect at some nominal alpha equals 0.05 level, which is a pretty standard thing to use. So should we use the estimates from our model with sex? Okay. So should we then start really just thinking about the one model, the model with sex differences versus the model without? So in the standard kind of alternate hypothesis, null hypothesis thing that you learn in basic statistics, that's exactly what you do. You accept the hypothesis, you reject the null, so you then need to start focusing in on the model that you accept. So one wins, one loses. But is that the right thing to do? Well, here's the problem. If you don't do an experiment, which is most of the time, all right, then you have a situation that you're going to have to wrestle with. So let's go back to that sort of spatial difference in this alpha parameter thing, all right? So let's say you're really interested in whether alpha varies over time. So remember what we did. We looked at a model with alpha sub I, variation over space rather, and a model alpha dot, no variation over space. So if I look at this model, the one I'm circling here, let's use our fancy laser pointer. So what do we have? Here's alpha sub I versus alpha dot. The beta parameter is beta sub I and beta sub I. So this model and this model are nested. This model here that I'm now circling is the one that's nested here. This is in fact the one that we introduced a few slides ago. So comparing the likelihoods of this model and this is a test of whether or not alpha sub I does better. It's a test of whether there is a spatial variation in alpha. But what about these two models? So here's a model that's got alpha sub I compared to alpha dot, but the beta parameter is beta dot in both cases. In either of these two situations, both of these are tests of alpha. This model is nested within this. This model is nested within this. Okay? So that's a test of alpha sub I versus alpha dot, but so's this one. And in fact, there's an infinite number of possible models that you could build where one of the models is alpha sub I and one of the models is alpha dot, and comparing the two is a test of alpha, but all the other bits of structure in the model are going to influence the results you get. Okay, so here's an example of this drawn from the data that were underlying this alpha beta model. So here's two models, alpha, I guess I have a time here, really should be the I. 7 parameters, there's negative 2 to the log likelihood. Here's the dot. All right, 5 parameters. There's the likelihood 839. Again, we see that the model that's bendier, has more parameters, fits a little bit better That likelihood is smaller. 2 degrees of freedom, chi-square 1.16, but there's the p-value. Now, I do the exact same thing, but now instead of fitting models with variation in beta, I make them dot models. Now, what do I have? Well, I have basically the same sort of problem, except now I've got a chi-square that's strongly significant. And so one says no difference, and one says difference in alpha. Which one do you pick? Test of time effect over here says alpha is not important. Test of time effect or spatial effect over here says it is important. Which one do you use? And that's the problem. If you don't do an experiment, you're stuck. And what's another problem? Well, the other problem is that not all models are in fact nested. Okay? Not all models are nested. For example, if you have a model where some parameter is a function of rainfall and some other model where the same parameter is a function of altitude, well, altitude is not a special case of rainfall, and rainfall is not a special case of altitude, so they're not nested. All right? So how do you actually figure out what to do? And the answer is we need to think about something that doesn't involve classical p-value based hypothesis testing, and that's the subject of model selection, which is what we're going to cover in the next part of this lecture. All right, so take care, and we'll reconvene to talk about model selection next time.